Yep, go ahead.
Great. Thank you everyone to come to today's Immune Zoom seminar. It is my absolute pleasure to introduce today's speaker, Dr. Taha Merghoub, who is currently a professor at Weill Cornell Medical College. Taha received his bachelor degree from University of Algiers and a master and a PhD degree with the highest distinction from the University of Paris. His thesis work focused on the study of genetic polymorphism in fetal hemoglobin genes in patients with sickle anemia and thalassemia. After that, Taha came across the Atlantic Ocean and did his postdoc with Pier Paolo Pandolfi at Sloan Kettering. And since then, he stayed in the New York City area. During his postdoc, he characterized the transcriptional properties. During his postdoc, he characterized the transcriptional properties of the ZBTB7 gene and its role during development. After that, Taha started, I think, a phenomenal research career on cancer immunology when he partnered with Alan Houghton and later Jed Wolchok at Sloan Kettering. In 2022, Taha becomes the Margaret and Herman Sokol Professor in Oncology Research and the Deputy Director in the Sandra and Edward Meyer Cancer Center at Weill Cornell. His lab focuses on developing novel cancer immunotherapies. He studies the pathogenesis and treatment of melanoma and developing tools to understand melanoma and multiple other cancers. Taha has made numerous influential discoveries on cancer immunology. For example, his lab found that the effect of CTLA-4 blockade on the destabilization of Treg cells, it depends on Treg cell glycolysis, which suggested that decreasing tumor competition for glucose may facilitate the therapeutic activities of CTLA-4. More recently, his lab revealed an unexpected role of neutrophils in eliminating tumor antigen escape variants. This study demonstrated an exciting interplay between T cells and the neutrophils in anti-tumor responses. So without further ado, Taha, the floor is yours.
Thank you very much for this very nice introduction. I am going to share my slides now. Can you see my slides?
I think if you can play the full mode, yes, I think now it's great.
So do you see the regular mode or presentation mode? So you see one.
Actually, right now it's a regular mode.
I think it's perfect.
Yes.
Okay, perfect. So it's very nice to be with all of you today, and it's a great pleasure to spend some time with you today and share some of what we do in the lab. And the first thing I always like to do is to think of what's happening between today and tomorrow. And for this reason, I'd like to wish a happy Father's Day to all the fathers who are here on the call with us, and for tomorrow, happy Juneteenth. And I think it's great that we're now celebrating that day. Those are my disclosures. And so I just wanted to, since I just heard you talk about my transition, and I just wanted to highlight that, in fact, after 24 years at Memorial Sloan Kettering Cancer Center, and I like to show this in black and white because it feels that I've been there forever, I actually moved from Memorial Sloan Kettering Cancer Center to Weill Cornell Medicine, and it was a huge move because I moved from 69th Street South to 69th Street North. I actually see my other building from here. from my current building, I can see my old office, which is quite funny that by just changing my street, I changed quite a bit of my life. And I am very happy that I joined the Department of Pharmacology there, led by Lorraine Gudas, and actually I'm acting as Deputy Director together with Jed, who is the new director of the Mayer Cancer Center at Weill Cornell Medicine, and it's a great new adventure. And so just a quick outline. And so today I'm going to give you a quick introduction on the rationale of the works we do within our group. And then I'll tell you about the role of innate immune cell as a factor for immune-based therapies, which is our recent discovery, which shows, a specific role of neutrophils in tumor elimination. And finally, if we have time, I'll briefly touch on some of the work we have done on double positive cells, which was published last year, but I think was not very well advertised, but I think it's quite an interesting finding. And so just as an introduction, when we think of targeting cancer, right, we are targeting to means to target tumor cells directly, such as with surgery, chemotherapy, radiation therapy, and mainly targeted pathway inhibition. And of course, there are indirect means of targeting the cancer cells, which is using immune-based therapy, but we can think of many other approaches which will affect our immune system, such as, for example, changes in nutrition or lifestyles, etc., etc. And so melanoma, which is the model system on which we work, and again, I insist it's a model system on which we work. We care about many cancers, and so melanoma was considered the poster child of two main paradigms in cancer treatment. One is targeted therapy, and the other one is immune-based therapies. And so when we think of melanoma and we do a fast forward to the past, right, we realize that in fact, if a patient came in 2010 and asked for, what therapies are available, then the pictures were quite grim. You have mainly two treatments, which is the TTIC chemotherapy and high dose IL-2, and maybe 5% of survival rate, 10 to 5%. And however, if we look at the landscape in today's world, right, at the present, we see that many more therapies have been approved in melanoma. One of the latest one is the tebentafusp, and I'm showing in red all the immune-based therapies, and of course we have all the targeted therapies. And you see that thanks to our work in research and translational research, we have made few advances, and the picture has changed quite a bit. And so if we're thinking of melanoma, maybe we should just touch on what melanoma is and how it shows up. So this is just a little schematic where you see the epidermis and the dermis. And between the dermis and the epidermis, you have what we call melanocytes, which sit here between the dermal and epidermal junction in human. And so when you have oncogenic mutation, generally you'll have a proliferation of these melanocytes, which are sitting in the dermal epidermal junction and form what we call benign nevi. And if any one of us look at their skin right now, we'll see that we have this little nevi, which probably have an oncogenic mutation that's mutated. However, that one event is not enough, and things don't move. However, as you accumulate other genetic alterations, you see there are changes, and you go from this plastic nevi to the radial growth phase, vertical growth phase, and then you become metastatic melanoma. And actually, that's what really makes the melanoma deadly. And that's what gives the bad name to melanoma. And when we think of the important factors in melanoma, this is just a little cartoon. This is the extracellular membrane. You see that the RAF MAP kinase pathway is generally mutated in melanoma, not always. And you have another mutation which is associated with mutation in a tumor suppressor gene, such as P10, for example, and understanding this molecular mechanism which are associated with melanoma led us to, developing a personalized medicine in melanoma. And as an example, you can see here, different melanomas, some are ranging from screen with chronic sun damage to the, the acral melanoma, which arise in non-exposed skin, uveal melanoma also, non-exposed to sun. And you see that for each subtype, there are different mutations associated with it, right? And so that led, of course, to the development of therapies which will block only the BRAF pathway, or KIT, for example, and that will lead to this targeted therapy concept. And of course, this is all great and nice. When you have these therapies, you are basically targeting oncogene or tumor suppressor gene, mainly oncogenes, and there you're targeting tumors directly. However, of course, there are many other factors which are important in the generation of this melanoma, such as interaction with immune cells, trauma, and blood vessels, and any of these are a good target. And of course, in our group, we are interested in targeting immune cells. And what we try to do, of course, since is trying to find a balance between, eliminating cancer cells as opposed to generating an autoimmune disease, and just putting it in a little cartoon, trying to be a little funny here, is that in fact an autoimmune disease that's associated with melanocyte is the resurgence of vitiligo or depigmentation, because your immune system is attacking melanocytes. So what we try to do is do the same thing and mimic what we do, but in fact attack the transformed cells, meaning the melanoma itself. And there we are mainly basically trying to target the mutated self, and we have quite a bit of work in this area, and I'm not going to touch on this today, but I'll do something else. So as I showed you before, melanoma is happening through multiple genetic changes throughout time, and the way we can think about this is at initial stages, the immune system at the same time evolves, right? So when a mutation appears in your in melanocytes, in general, the immune system will recognize it either by CD8 effector cells or NK cells, and with, of course, the help of CD4 and antigen presenting cells. However, when we get to the escape phase, which is the cancer itself, you have multiple events which are associated with the preventing these, immune machinery to eliminate tumors. And we can count, for example, myeloid suppressor cells, changes in NDCs who become tolerogenic, for example, T cell being exhausted by expressing exhaustion markers, Tregs suppressing this, or metabolically by expressing IDO, for example, IDO, for example. And so really what we try to do, right, is reverse that effect and restore this elimination phase of the immune system. And I think just showing this slide, of course, that doesn't do justice to all immune therapy, but what I'm trying to show here is that immunotherapy is great, or immune-based therapies with immune checkpoint blockade are great, because when we look at this is survival of patient, this is the first patient treated with ipilimumab or anti-CTLA-4, you see you have a long-lasting response. However, not all the patient respond. So it's hard to predict who responds, but once you have a response, in general, you have a long-lasting response. And that's really an important characteristic that we want to use. And so the goal, of course, of many in the field, not just us, is to make sure that we have more responders than just a low number of responders to immunotherapy. And some of the question we are asking in the group is, can we predict response to immune therapy reliably? And can we improve response to immune-based therapy? And so when thinking of this question, So typically cancer patients will all show up with full-blown cancer, basically, and so a small subset of patients will be cured, but a big subset of patients will be resistant. And when we think of why they're resistant, you can have tumor intrinsic mechanism, meaning changes in the tumor cell itself. And we have done few studies looking at tumor neoantigen or mutation antigen and how they're seen by the immune system. You can have mutation in the antigen presentation machinery, meaning the T cells don't express MHC class 1, and not the T cell, the cancer cell don't express class 1 anymore, for example, and they're not recognized by immune system. But there are also the immune mediated resistance mechanism, and this is where I'm going to go today. And so I'm going to focus on immune mediated resistance mechanism and look at that. And so when we are thinking of that, this is a picture from one of Kayevsky's paper. In general, you have patients who do not respond because the tumors are cold, so you don't have immune cells in them at all, or you have patients which respond very well and they are hot tumors, those are T cells, but then as a compensatory mechanism, what happens is that you have expression of immune resistant of suppressive cells such as Tregs, expression of PD-L1, for example, or IDO here. So those, if we think of the cartoon I've shown, right? So those are among the mechanism which will balance and block a response. And so when we are thinking of this in a simplified way, and I know this is very cartoonish, so think of cold tumors, hot tumors, and so when we think of the hot tumors with pre-existing immunity, the reason why they're not responding anymore is that there is a suppression mechanism that have been in place. And when in some of the patients we have looked at a while back, actually, is that you see that all the patients that responds, they have an interferon, a type 1 interferon signature, however, to the one that respond to anti-CTLA-4 and the one that do not respond do not have this type 1 interferon signature, suggesting that these have the pre-existing immunity and these don't. And so if we made this like super simplified, right? So patient with pre-existing immunity, if we add immune checkpoint, it's enough. And the ones that don't have pre-existing immunity, you need to bring immune cells to the tumor and then add the immune checkpoint. And the question is, A, how you remove suppression, and B, how you go from here to here. And those are things you are trying to answer with the following 4 points in the lab. So in the lab, we try to remove immune suppression, and we have some work on blocking myeloid suppressor cells, Tregs or IDO, for example. We have work on metabolism that was presented and suggested at the introduction. We work on boosting existing T cell response. And so although a lot of people have given up on co-stimulation using GIR and OX40, we're still studying that. We think that the problem is not the molecule themselves, is the way we have designed clinical trials, and that should be looked at more carefully. We're trying to force antigen presentation and finally induce or prime T cell responses, and we do that with immunization, in situ vaccine, or supplementing the T cell repertoire with CAR T cells, for example. And so today, what I'm going to do is focus on this, because I have, I think, an interesting story which is quite recent on boosting the T cell response and what we found for the innate cells. So when thinking of immune modulators, you have a molecule that inhibit inhibitory receptors, such as CTLA-4, which is approved, PD-1 approved, and LAG-3 now has joined the group, right? And of course, there are other candidates, but there are also co-stimulatory molecule or activating receptors, and those are quite good targets, provided we do we do our clinical trial based on science and not just based on the interest of a pharmaceutical company. And so one of the molecules we are looking at is OX40. And why are we interested in OX40? OX40 is a great molecule because it actually, when engaged on CD4 T cells or CD8 effector cells, so CD4 effector cells and CD8 effector cells, it increases their proliferation, clonal expansion, cytokine production, and it promotes their memory T cells. And the great thing is that at the same time, it deactivates suppressive cells by eliminating them through multiple mechanisms, including AICD and through Fc receptor elimination. So we studied this molecule in the context of melanoma, and we've seen that as a single agent, it can remove small tumors, but it does not remove, aggressive voluminous tumors. And so what we have done with Daniel Hirschhorn in the lab is we've devised the treatment where we give cyclophosphamide, which is a treatment which will contribute in the killing of tumor cells, but also will create space by generating lymphodepletion. And then after that, giving antigen-specific T cells. So this is adoptive T cell therapy with CD4 T cells, which are specific to a melanoma antigen expressing B16, and the addition of this co-stimulatory molecule, which is anti-OX40. And after 3 weeks, you see that these tumors regress completely. And so using this model, we have shown using a transgenic mouse, which express where melanoma is driven by the metabotropic receptor. And this is in collaboration with Susie Chen at the time. You see these mice all have tumors in the snout, ears, etc. And when given the therapy, the combination therapy, when untreated, you see that the mice, this tumor increase, and it's quite ugly actually. However, if we are giving this therapeutic regimen, you see that the mice actually lose tumors, but also depigment, because you are also getting rid of the normal melanocytes. And so the interesting thing with this model is that not only we had depigmentation, which is what has been described with this with TRP1 cells in the literature, we also have another special immune adverse event, which is increasing the thickness of the ears and the snout, and everywhere where we have melanocyte in the dermal junction. And it was a great model to actually look at immune adverse event just by measuring the ear thickness. And so looking at this model, we actually asked the question about the effect of heterogeneity in tumors. So the question we're thinking about is, if tumor are heterogeneous, is targeting a single or subset of antigen an effective strategy? Right here we are targeting a single antigen, which is TRP1. So are we able to eliminate heterogeneous tumor when you do that? If so, do we need to target every clone of tumor for tumor elimination? And can we mobilize the endogenous immune system to eliminate heterogeneous tumor? And when we did that, we were thinking basically that we will adoptively transfer antigen-specific T cell, but then the endogenous T cell repertoire will maybe eliminate the antigen escape variant. But we were surprised by saying something different, and I'll tell you in a minute what we saw. So basically, if a tumor is homogeneous and you're targeting one antigen, the thinking is that everything will be eliminated. However, if you target a tumor that is heterogeneous, then you will have a subset of cells that will resist the elimination, and then after a certain time, they will repopulate the tumor and will have an antigen escape variant. And so we tried to see if we can model this with our system and whether it's relevant to human. And when we took a bunch of patients, those are those are melanoma tumors from different patients, and we look at TYRP1 as an example, which is a melanoma differentiation antigen. We see that some patients, this is one patient, express TYRP1 in 100% of their cells. We see that some patients express TYRP1 only in a subset of cells, and other patients do not express TYRP1 at all, suggesting that you'll have different presentation of this antigen depending on which melanoma you're looking at. And then we actually looked at the bigger series, analyzing this data from the single cell analysis from Nirna Cohen's group, and we see that when looking at TYRP1, tyrosinase, DCT, cancer testis antigen, so different antigen, we see that there is some heterogeneity in how they present or express this antigen. And when we look at TYRP1 again, you see that patient 1, patient 2, patient 3 as an example, you see that some do express it, some have heterogeneous expression, some don't express it in most of the cells. So using the model that I've shown you just now, what we tried to do is to mimic this heterogeneity. So what we did is we take B16 cells or B78H1 cells, which are B16, which do not express TYRP1. So, and with the antigen loss. And what we show is that when we implant the mice with these tumors, if we treat them, the one that have the antigen will regress completely. The one that don't have the antigen will grow. And what we did to mimic this, we actually implanted the tumor in one side without the antigen, the other side with the antigen, and we see that again, the tumors regress when they have the antigen, the other one don't regress. However, if we mix the tumor, this tumor with this tumor in one flank, we see that surprisingly everything regresses, right? Suggesting that there is another mechanism that leads to the elimination of the antigen loss variant of the B78H1, and it's only within the tumor. And so trying to understand how that happened, what Daniel did is we tried to image the flux of the adoptively transferred T cells, and what we see is that the adoptively transferred T cell come in and then leave quite early. However, the tumors continue to regress, suggesting that there is another cell type that may be responsible for this. And then what Daniel did is to actually deplete CD4 T cells, which are the ones that are helping for this tumor elimination in this context. And so if we remove the T cells early, the tumor protection is lost. However, if we remove the T cells later, we see that the tumor protection is not lost, but we have a different effect on the immune adverse events, which are shown here. And so we're thinking so maybe what we're looking at is not all based on T cells. And so what we did is we took extract from these tumors and tried to understand what cytokines are made in this tumor. And what we see, interestingly, is that it's mainly innate cytokines such as the MCSF, for example, MIP1 alpha, MIP1 beta that are made, which is not what we think of when we're thinking of what T cells are making. And so we're thinking maybe we're looking at the wrong thing then. And so what we did subsequently is we tried to characterize which other cells are different in these tumors. And what we see is that there is huge infiltration of CD11B and LY6 cells, suggesting that neutrophils are quite important and they are dominant in the treatment group. And we tried to look at something which is related to one of the therapies, which is approved in clinic. And so we looked at anti-CTLA-4, and we see the same thing, but at different extent. And as shown here, this is quantified. And you see that there is an increase in both CD11B and LY6 in this context. And of course, there, and I'm not showing you all the data, we started scratching our head and thinking, okay, neutrophils are important. But then when looking at the literature with neutrophil, it was quite confusing because most of the literature described neutrophils are as important as pro-tumorigenic and not anti-tumorigenic. And so one of the things that we have thought about initially is about netosis, thinking that this that neutrophil can make neutrophil extracellular traps or nets as a mechanism. So we know that they have that as a mechanism of tumor elimination, and so the idea is that the neutrophils will give these nets, which are nuclear membrane associated nuclear membrane disruption, and there is pad citrullation and chromatin decondensation that will then be expulsive and eliminate pathogens. And so we're thinking that maybe that is what is helping remove tumors. And so at that time we connected with Michaela Egelbald, who is very recognized as somebody who has expertise in this area because we didn't know it. And we actually looked at nets, and interestingly, we saw that by staining for DAPI, myeloid peroxidase, and citrullinated histone H3 that there is an accumulation of NETs in the tumors of these mice. And the same thing was done for anti-CTLA-4, and we saw that there is an increase in neutrophil and an increase of NETs in both settings, suggesting that there is an association between tumor elimination and NETs. However, of course, this is not proving that there is a causal relationship. And so in order to look at the causal relationship, what we try to do is actually deplete neutrophil using anti-Ly6G. And when we did that in homogeneous tumors, meaning only B16, we saw that tumors were still eliminated, and most of the mice were cured except this exception. And when we did mixed tumors, as the one I showed you before, we saw that in fact, if in this context we depleted the Ly6G, then we didn't have protection anymore. And now we see that elimination of the antigen escape variant B78H1 or the B16 without TRP1, we don't have the same phenotype anymore, suggesting that neutrophil play a role in the elimination of the non-TRP1 positive cells. And so then we went through the netosis route, and we used PAT4 knockout because PAT4 is important for netosis, and we see there is no effect. We looked at the elastase, and there is no effect also. So and then finally we realized that iNOS is important in this effect. So and so if we use iNOS knockout, we see that we lose the protection given by the neutrophils in this context. And so those are full knockouts, and the effect might be indirect. So in order to see whether the effect is associated with absence of iNOS in neutrophil, we took neutrophil that we co-cultured with B7H1, and we see that wild-type neutrophil are able to kill these tumors. However, if iNOS is not there, the killing is at a lower extent, suggesting that iNOS is playing a role in the killing of tumor cells by neutrophils. And so looking at the time, I think I'm still good. So and so then, of course, we did what everybody does. What is so specific about the neutrophil after anti-OX40 treatment and cyclophosphamide treatment? And so we saw that, in fact, when we did the PCA of whole transcriptomics, that the neutrophil in the treatment group versus the IgG cluster completely differently. And in order to understand that better, we did site-seq from mice treated with TRP1, OX40, or IgG as a control. And what we saw is that, in fact, in the treated mice, there is this cluster 1, which accumulate, and that is different from the IgG treated group. And so this cluster 1 was associated with better response and anti-tumor response. And so we then trying to understand what is this cluster 1, why is it, what is special about cluster 1, and we see that the difference between both is that cluster 1 has neutrophil which are more mature than the neutrophil in the untreated group, and it's associated with signature for neutrophil migration, chemotaxis, cellular response to chemokine, etc., suggesting that really this group is quite different. And when we look here as an example, this is cluster 1. You see that cluster 1 is associated with chemotaxis, neutrophil killing, apoptosis, necrosis, which we think is just a biomarker of that. And the other cluster, which is not increased in cluster 1, is you have marker of granulation, azurophil, and gelatinase, which is associated with basically younger neutrophil, if you will. And then we try to understand the relationship between the two type of neutrophil and whether one was coming from another. And we did this trajectory analysis with David Redmond, and we show that really these two trajectories are independent. And so we see that initially they all start here with these markers of early neutrophils, and then the one that are in cluster 1, which is the neutrophils that are able to kill our antigen escape variant. We see that you have expression of certain chemokines such as 6CL3, CL4, and CEBP beta, which is associated with more mature neutrophils, and so showing that these have a very specific signature. And then so then we looked at the translational potential of this of these anti-tumor neutrophils, and what we did is that we actually went to the TCGA and compared the signature from cluster 0 and cluster 1. Although I'm not a big fan of this type of analysis, we still did it. I don't think it's necessarily conclusive, but I think it helps you maybe know that this is relevant in the clinical context. And you see that cluster 1 is associated with melanoma patients which do better in the TCGA. And we looked actually at the clinical trial with Andy Weinberg of patients which were treated with anti-OX40, and they were just given 1 dose of anti-OX40. And we see that in this case, looking at pre and post biopsies, that in most patients, you have actually an increase in netosis or nets, which we consider here as a surrogate marker for neutrophil activation. And so then one of the questions that we had is, what is this relevant not in the context of adoptive T-cell therapy and on one therapy? And so we took MC38 mice, which we treated with anti-OX40, and because we know that they respond to anti-OX40, and we depleted neutrophils. And in that context, we see that actually depleting neutrophil reduces the efficacy of the treatment with anti-OX40 as single agent. We've done the same thing with the same model, but this time treated with anti-CTLA4 and anti-PD-1. And you see that, again, you reduce the efficacy of anti-CTLA4 and anti-PD-1 given as, dual agent, actually, but not in the context of adoptive T-cell therapy. And then we looked at patients' biopsies that were treated with immune checkpoint, and we see that in general there is an increase of nets in patients which are treated with immune checkpoint blockade, as shown here for 3 patients, but you see that it is the case for multiple patients. And then with David always, we went and looked at the series of patients which are treated with anti-PD-1 or anti-PD-1 and anti-CTLA4, for which there were biopsies before treatment and after treatment. And what we see is that in patients that were CTLA4 experienced, there was a difference between the ones that had the signature that we found in mice of the cluster 1 and the other one, suggesting that maybe in patients, this signature is also relevant and neutrophil play a role. And so this is just the model that we came up with. What we think is that T cells, when engaged through either OX40 in this context or anti-CTLA4, anti-PD-1, will produce pro-inflammatory cytokine and chemokine, which will recruit and activate neutrophil, which will in turn have some tumor killing directly through this inos, and these neutrophil will kill tumors indiscriminately of antigen production. And if you want to read more on the detail, this was published in Cell in March this year. And so then one of the questions we are asking right now, and I'm not going to show you the data today, is given what we've seen as immune adverse event, we think that nets are also important, or these neutrophils are so important in immune-related adverse event, because we looked at patients and in their nets and we saw that. So, and quickly, I've shown you this before, that we have this ear thickness, which is full of nets. This allowed us to measure the nets, this side effect in mice. we see that there is increase in neutrophils in mice which have this side effect, and we see that interestingly the T cells go away and neutrophil come in as this immune adverse event is showing up, and when we get rid of the neutrophil, we actually we can, if we look at the ear thickness as a measurement, we can get rid of them, and as shown here in this graph. And this is associated with netosis. We went into patients, we see that there is netosis as an immune adverse event, and I will ask everybody if not to basically share this part of the data of the presentation, because it is unpublished, and you see that this patient will actually present much more nets and when they have rash. It's not always the case, but most of the time they have this net, suggesting that neutrophils are important. So we think that maybe the immune-related adverse events are associated with the presence of the neutrophil. Now, whether it's through INOS or through nets or other mechanism, we don't know that yet. And so, and this is ongoing. And so, again, so we think that when you have normal skin and you recruit T cells to the skin, you probably provide this inflammatory cytokine, which will either, so the T cells will kill melanocyte and generate depigmentation, but if they produce this inflammatory cytokine that recruit a neutrophil, then you will have this immune adverse event that I've shown you. And so in the future, we'd like to define the difference between pro-tumorigenic and anti-tumorigenic neutrophil, characterize the neutrophil and contribution in patient treated with immune-based therapy, and determine the contribution of neutrophil in immune adverse event, and hopefully find biomarkers for this neutrophil population. And so I think I have maybe 4 more minutes, if I'm correct. Yes? No? Hello?
Yes, you have a few more minutes. Yeah.
Okay. Okay. And so I just have 3 slides or 4 slides on this story that I call "Don't Ignore the Biology You Don't Know or You Don't Understand." And this is just a quick story that we have found also serendipitously. So if you think of T cell development and the commitment of single positive fates, you think what we learn in our textbook, right? We see that T cells will acquire as they mature, they will acquire this double positive phenotype, and they will be CD4 positive and CD8 positive cells. And then after positive and negative selection, you will have CD4 single positive cells or CD8 double positive, CD8 single positive cells. And this is how we have been thinking about, right? So the T cells with weak interaction will survive, and the one that have no interaction will use cell death. So and so the way we've been thinking when we see T cells which are double positive in the periphery, we're thinking of this as being cell doublets, which is just, you know, an artifact. And we're thinking of cells which underwent trogocytosis or membrane exchange. So this is why we have double positive T cells, or they're escapees from thymic selection. However, what we've seen is that, in fact, in melanoma, in mice and human, you have double positive T cells. And we saw that these double positive T cells mainly accumulate in tumors. And if you see the imaging here, is that these T cells are singlet, they're not phagocytosis, they're just cells which express both markers, CD8 and CD4s, and they're different from the other cells. And so we actually characterize these cells, and we realize that, in fact, these cells are changing in the periphery. So CD4s which become highly cytolytic, actually will acquire a CD8 marker, and they will become CD4 and CD8 double positive T cells. And these cells are highly cytolytic and high killer cells. And what we also found is that CD8, which acquire suppressive capabilities, become CD4 positive as well. So we'll have CD8 and CD4 positive T cells. So this CD8 and CD4 positive T cells, if you look, they express FOXP3 and they become Treg-like. And so now when we're thinking of double positive T cells in the periphery or in the tumors, we need to think of at least 3 functions. You will have the naive CD4, CD8 double positive T cells. We'll have the highly cytolytic CD4, CD8 double positive T cells, and the regulatory CD8, CD4 T cells. So, and I invite everybody to look at this. This was published in JAM last year. And finally, I would like to, of course, the work I presented is not something I have done on my own. It was done by, multiple talented people in the group, and I'm very happy that. And I'd like to mention Jed Walchuk, who is my partner in crime, Daniel Hirschhorn, who mainly developed the neutrophil story, Lucas, who actually is looking at immune adverse events in patients and in animals, and of course, everybody in the group. Thank you. And of course, our collaborator and mainly the people who help us understand what I don't understand, who are our bioinformaticians with whom we collaborate very closely and help us understand a few things. So I'd like to thank everybody. And of course, I welcome any connection through email in the future. This is my email. And I'm happy to take any questions. Thank you.